Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Okay, hello everybody, welcome back to another episode of the A-Level Cookbook. Today we're going to be doing the AQA A-Level Biology Paper 2 from 2018, and this is the exact paper that I did as well, so it's going to be interesting to go back to. So it says heat stress is a condition that often occurs in plants exposed to high temperatures for a prolonged period of time. Heat stress is a major factor in limiting the rate of photosynthesis. So heat rate decreases the light-dependent reaction of photosynthesis. Explain why this causes a decrease in the light-independent reaction. So they've told us in the question we're going to get less light-dependent reaction, and then they're also telling us as light-independent reactions dropped. We need to say why. So the whole point of the light-dependent reaction is to make ATP and reduced NADP. Because in the light-independent reaction, the Calvin cycle, RUBP is combined with CO2, and you get two lots of GP. Those two GPs are turned into TP, and in that process, what's happening is reduced NADP is oxidized to become NADP, and the GP is reduced to TP, but this also requires ATP. So ATP becomes ADP and PI, and so forth. These two come from the light-dependent reaction. The entire point is to try and get that. So here, it says explain why there's a decrease in the light-independent reaction. It's because you've got less ATP and less reduced NADP. That is all. Another effect of heat stress is a decrease in the activity of the enzyme Rubisco. A decrease in the activity of an enzyme means that the rate of reaction to catalyze is slower. A decrease in the activity of the enzyme Rubisco would limit the rate of photosynthesis. Explain why. So it's saying Rubisco no worky, why photosynthesis no worky? So what you're going to say is exactly the role of what Rubisco is. That means you're combining less CO2 with RUBP, which means you've got less GP, and then less TP, and so forth. So scientists investigated the effect of temperature on the activity of two enzymes isolated from the leaf cells of cotton plants. We've got Rubisco and Rubisco activase. So an enzyme that activates Rubisco, and figure 1 and 2 show their results. So we should always look at the dependent and our independents. So dependent, independent, dependent, independent. So for each of these, the independent is the temperature, and their dependents are what? Rubisco activity here, and then here it's Rubisco activase. And then have a look at each data sheet or thing that they give you, and just take a quick look at what's happening, and get just familiar with what's happening. So if we look here, we can see Rubisco activase is here, and as you increase the temperature, the activity of it is going pretty high at 40 to 50. So between those two. And beyond that point, we don't really know what's happening. Maybe it goes down, we don't know. It's only up to here. And then Rubisco activase, what happens is it starts to go higher and higher and higher, and then it peaks at about 30, and then it goes back down at 60. Interesting. So the scientists concluded that heat stress reduces the activity of Rubisco in plants by affecting Rubisco activase. Use this to evaluate the conclusion. So they're saying that Rubisco activase, so heat stress, does something to Rubisco activase, which then for some reason reduces Rubisco. So let's have a look, shall we? And it says when you're evaluating as well, you need to say what supports their conclusion. So supportive things. What directly contradicts it and why? So you should describe it, say the data shows this, what could this possibly mean? And then as well as that, validity. So validity problems are basically, can I take this and apply it to absolutely everything else in the universe? So you look through each step, each sentence, and critique it and say, does this link to their conclusion? Their conclusion is saying that if you do this heat stress stuff, Rubisco activity activase changes and Rubisco activity drops. And that's in plants' leaves. So let's have a look. So if we start with our supportive factors, if we go back to a thing here, let's see what supports what they're saying. So it says here that heat stress reduces the activity of Rubisco in plant leaves by affecting Rubisco activase. So we should look really at what's happening with Rubisco activity and where it's reducing. Well, I'm not really seeing a decrease because you can see Rubisco activity is going up, but it is going up at the same time as Rubisco activase, which supports that these two may, be dependent with each other. This may cause this to go up, right? And if we look here, up to about 30, this is increasing, and up to about 30, it's also increasing here. So that's something that does kind of support what they're saying, because if we're increasing and the other one's increasing, then surely that's there may be a link there, and if they decrease, then they decrease, right? So then you would say that. So something that could support it is that Rubisco activity increases up to around, what is that, but we need to remember, I'm only focusing what's between these two, increases up to 30 degrees C with Rubisco activase, right? But then beyond this point, this is still going higher, but then this is going lower. So that's kind of contradicting because it's like, why would that be the case? So if we, like Rubisco activity still increases after this 30 mark, but Rubisco activase decreases. Therefore, it must be that the Rubisco activase's optimum temperature, we'll write just RA for short, is around 30 degrees-ish. You write the whole thing, I'm just being short here. And there's not really much else that's supporting it, because if this is going up, and if, the Rubisco activase activity is also related to it, then surely if this goes down, this should also go down. But you can see, beyond that 30-degree marker-ish, it's still going up, despite the fact this is going down. So that's directly contradicting what they're saying. So, it's still increasing optimally, but then also it's the Rubisco activity is still increasing despite a decrease in Rubisco activase. You need to see what the possible thing could be that you can conclude from that. This suggests that Rubisco activity doesn't affect Rubisco. It's still able to go to town and keep going higher, right? So when you're talking about it, you need to sort of like say, what the possible links are with these two, right? What supports it here is that, again, up to that point, they're both increasing. What contradicts it is that for some reason, even beyond this point, it's still increasing, even though we've passed the optimum for Rubisco activase. And that suggests that there could be, like, for example, it suggests that this doesn't actually, affect the activity of Rubisco. So then we need to talk about validity points. I'm just going to, validity is basically, am I able to take this and apply it to every single thing in the entire universe if I were to do it somewhere else? So you should go in the mind thinking, okay, if I was in a different center, a different place, and I did this, would it work? And what would I need to know? So what I mean by that is if I come up to you and I did a trial and I was like, all right, 10 people in a room, I gave them a Greg's sausage roll, and all 10 people shat themselves. And I said to you, all right, I did the study, and 10 out of 10 people shat themselves after eating a Greg's sausage roll. I can conclude that 100% of people will shit themselves after a Greg's sausage roll. And you'd be like, no, or so I hope. And then I'd be like, why? And you'd be like, well, I mean, you did get 100, but I'd be like, I got 100%. I'm like, yeah, but, and then you'd start critiquing. You'd say, oh, you've only done it in 10 people. You've only done it with, we had no idea what these people are like. We don't know if they have any allergies to anything. We don't know this or that, blah, blah, blah. You've this and this. And you wouldn't go and then say every single person in the universe who eats a sausage roll will shit themselves. So the same sense, it's the same thing here. Validity is that. So we're going to go through it step by step and see what's up. So what they did is they looked at the temperature and the activity of two enzymes in the leaf cells of cotton plants. Fine. But then their conclusion says it's the activity in plant leaves by affecting them. You can't go ahead and conclude that for every single plant leaf in the universe when you've only tested it in leaf cells of cotton plants. So already, it's only done in leaf plants. It's not really valid because what if you do something, of cotton plants, leaves of cotton plants, because it's not really valid because you can't, can I do this with other leaves from other plants? And not just that, look at what they're measuring as well. So they're dependent is Rubisco activase activity and Rubisco activity, other way around, right? But their conclusion is saying that this, if you heat stress leaves, in plant leaves, this happens. They've only tested it in isolated enzymes. They've not actually done it in an actual leaf. They've just taken these enzymes out. What if it's different within the leaf? As well as that, I have absolutely no idea if there's any statistical test done for this and whether they're significant or not, whether it is by pure chance as well. So that's another one to keep out for as well. When you're looking at things to do with experiments by scientists and stuff, you need to look for stats tests. Go look up any paper where they're measuring something in the entirety of on PubMed or something. Nearly all of them have a stats test of some sort. It's important because we want to make sure there is a statistically significant thing going on here, whether it's a difference in this or X and Y. So that's how I go through these, right? There are different types of gene mutation. Put a tick next to the box which incorrectly describes the effect of the mutation in an exon of a gene. So then, if we have a look, whichever one is incorrect. So a substitution may not result in a change in the encoded amino acid. That one is correct, so we're not going to do that one because you could remember that codons are triplets that code for amino acids. The DNA is degenerate, so maybe if you swap one, it might not actually have any change. An inversion will result in the change in the number of DNA bases. That one is the one that's incorrect because all you're doing is just reversing the sequence. It's not changing the number of sequences, the number of bases in it. So I don't know, you can have A, T, 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 A, C, G or something, right? If it's reversed, it would be G, C, A, T, 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 A. That's not changing the number of bases. Describe how alterations to tumor suppressor genes can lead to the development of tumors. Right, so this should be 3 marks because all you're doing is you got to describe how this leads to this. So you got to step by step it. And it's telling you alterations. So you need to think, what alterations do I know that can cause a problem with tumor suppressor genes. Remember, a gene is something that codes for a protein. If you're not expressing a tumor suppressor gene, you're not making a protein, so something is stopping that from happening. The other thing as well is it's talking about alterations to the genes. Acetylation is not relevant here because that's not being done directly to the genes. That is where you're doing things, you're acetylating the histones and changing them to make it either easier or harder to transcribe. So here, what happens is, imagine this is my gene here, like this is a section of DNA. If you slap on methyl groups like this, RNA polymerase can't get along and actually get in there and do what it wants to do. The whole point of any gene is the steps you've got to take is you've got to transcribe it and translate it to express it. Anything that screws that up is going to screw, its expression up. Another alteration is actually the gene itself, right? Because if you change the sequence of amino acids in it, you've now possibly changed the protein that's in it as well. So then the two things that I'm going to say is that increased methylation, what that does is it decreases RNA polymerase its ability to interact with the tumor suppressor gene. So no transcription and then no translation. That's my first point. And then the other thing as well is that you might have a mutation, which is any change in a base sequence. Mutation may lead to altered, amino acid sequence, which means you've got a different tertiary protein structure eventually, right? These are only just the mechanisms, but we haven't talked about how it leads to tumors. So that's what I mean by step, this, then this, then this, right? So the next step is to talk about how it leads to tumors. What happens is you get uncontrollable cell division, and that's what leads to your tumor. The whole point of tumor suppressor genes, it's in the name. Tumor suppressor genes make a protein that stops mitosis. And a proto-oncogenes, the way I think of it is proto means potential to, onco is cancer gene, which is the potential to become a cancer gene. So proto-oncogenes can become oncogenes. The reason being is because they stimulate cell division. So if you have a mutation that increases their activity or the expression, or they're expressed more, or they have a protein that works better, then you're going to lead to uncontrollable cell division, which leads to a tumor. So you need to step-by-step it. A type of malignant tumor divides every 8 hours. I'm starting with one of these cells. How many tumor cells will be present after 4 weeks? Assume none of these will die. So it divides every 8 hours. So we need to figure out how many hours we've gone through here. So 4 weeks, right? 4 weeks. That's going to be, so 1 week is 7. It's going to be times 4 of them, and there's 24 hours. So if we do that, 7 times 4 times 24 is 672 hours. So if one division is 8 hours, then 672 divided by 8 will tell us how many divisions we can squeeze into that. So 672 over 8 is 84 divisions. Now, rather than be like, oh, I know a formula for this, let's just use a bit of common sense, right? Like you start off with 1 cell, then you get 2, and then they double, and you get 4, and then you get 8, right? Each of these is 8 hours, as the question's told you. From here to here, you times it by 2, and then you times it by 2, and then you times it by 2, right? So we've had, 1, 2, 3 divisions. 3 divisions led to having times 2, times 2, times 2. So it was 1 times 2 to the power of 3. If I have 84 divisions, it's going to be 1 times 2 to the power of 84, surely. And that is exactly what it is. So you would do 2 to the 84, and you get a nice, lovely answer of 934 times 10 to the 25. Ideally, we should try and stick to the number of significant figures, or at least maybe one after, or something like that for what they've given us. So I'm going to just keep it as 1.9 times 10 to 5. There you go. Yeast can respire aerobically and anaerobically. A student uses the apparatus in figure 3 to measure the rate of respiration in yeast. So what they did is they positioned the flask in a water bath so the yeast culture could reach a constant temperature, and then they left the apparatus for 1 hour before starting their investigation. So let's have a quick look what's going on here. So again, you should always think dependent, independent, right? So independent, dependent. So they measured the rate of respiration in yeast. It's tempting to be like, oh, rate of respiration, but we need to know what they're actually looking at and measuring specifically, rather than just, oh, this and that. So they did this in a constant temperature, and they left it for 1 hour. Just have a quick skim to see what was up. So what they're doing is they're just looking at it over time. So the independent, I guess you could say, is time. I'm just thinking what they would plot on a graph if they could. The dependent is going to be how are they actually looking at this? They're looking at the movement of colored liquid. Yes, obviously that's technically speaking, the dependent is a rate of respiration, right? But they're using the movement of the color liquid to represent this, which might be useful to critique later on if they say, evaluate this, right? Or for example, if we're looking for something that needs to be controlled, we want to find whatever screws this up. So a question might say, I don't know, how would you control it? Describe what you would do. You'd look for anything that screws this up. For example, the temperature being inconsistent, or you know what I mean? Things like that. So here it says, suggest one reason why it was important that the student left the apparatus for 1 hour after the yeast culture had reached a constant temperature. Let's have a quick look here. Scale, capillary tubing, color liquid, yeast, and glucose. This is some stuff to absorb oxygen and glass tubing. So what they're doing is they're trying to starve these lot of all the oxygen possible, right? So that they're measuring anaerobic respiration, specifically by using the movement of color liquid to see what's up. So then if we come back to this question, suggest one reason why it was important they left it for 1 hour after it reached a temperature. Well, we want to starve it of oxygen. And if we have aerobic respiration happening, because now it's looking like they're trying to measure anaerobic, that's going to screw up the movement of the colored liquid. So what we need to do is make sure that there's aerobic only. So the only thing I can think of is that the oxygen, it's so that the oxygen is all used up. There might be some left over, but they're going to aerobically respire and use absolutely everything they can. Then what's left is they're going to just be doing anaerobic. That's the whole point of that being there. During her investigations, the colored liquid moved to the right. Explain why it moved to the right. So if it's going this way, then it means it's being pushed that way. So it means that these lot are making some gas that's producing it that way. Now, here, there's no oxygen, and they are yeast, so they must be doing anaerobic respiration. So already we can say, first of all, they must be doing anaerobic respiration. We need to think about what the difference is with anaerobic respiration versus aerobic in yeast. So in plants and yeast and stuff, what happens is you get glycolysis, which makes pyruvate, but that pyruvate doesn't go to the link reaction and doesn't do all of that. What happens instead is that, first of all, it's decarboxylated to make ethanol, so you get a CO2 out, and then that is reduced to form ethanol. And what happens is that reduced NAD is oxidized to become NAD, to regenerate it so that glycolysis can at least continue. So you get a bit of ATP out there. So the only thing that's like a gas that's been produced from here is CO2. So it must be that. So anaerobic respiration produced CO2. This still doesn't explain why it moved to the right though. There's an increased pressure in the flask, moving it to the right. The student found that the colored liquid had moved by this much in 24 hours. The diameter was this. The volume is pi r squared L, and that's that. Whatever. Calculate the volume of gas produced in centimeters cubed per hour. So we know that they moved 1.5 centimeters in 24 hours. So we think about it like this. Here's the tube. Here's the bubble that it's moved between here and here. That's going to be 1.5 centimeters in 24 hours, right? They've told you that the diameter of the hole or the lumen is 1 millimeter, and they want it in centimeters cubed per hour. So what I'm going to do is I'm first of all going to keep, that's fine, but I need to change this to centimeters. So 1 millimeter is 0.1 centimeters. Cool. The volume of a capillary tube is pi r squared L. Pi r squared L is now going to equal pi, what they want you to use pi is this. So 3.14 times r squared, which is going to be 0.05, because we want the radius, which is half of that, but we want it in centimeters, squared, times the length, which we worked out was 1.5. If we do that, we get 0.011775 centimeters cubed, but this is in 24 hours, as the question has told you. They want you to work it out in the centimeters cubed per hour, as in 1 hour gives how many centimeters cubed? We've done 24 hours, right? So if we want 1 hour, you're going to do 0.0117 blah, 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 whatever, divided by 24. If I do that, we get 4.906 times 10 to the minus 4, but we should try and stick to the number of significant figures the question has given us, which they've given us is 3 max. So 4.91 times 10 to the minus 4, and then, yeah, there you go. Figure 4 shows a typical population growth curve for yeast under laboratory conditions. So what we've got here is we've got a log 10 number of cells, and we've got time, and we can see that they start off all right, and then they shoot up, and then they get to here, and then they start dying, death phase, it starts to go down. The question says, explain why a log scale is used to record the number of cells. So the reason we do logs is because if we just plot this on a normal graph, immediately it would just look hideous and be gigantic. You'd need huge amounts of paper, because from here to here, for example, you might have gone up from 10 to a billion. So the reason we do that is because instead what we can do is we can write it in terms of the log values, which are just the powers, which means we can squeeze big ranges of numbers on the same scale. So the whole thing here is that we have a large range of values. Now it says many yeast cells die during the death phase, so just one reason why. So we need to think about this. What's going on is we've got the yeast cells are growing, they're happy, and then they live, and then they start dying. Nothing's changing within, what's being added and this and that. It's just a bunch of yeast in the laboratory conditions. So you need to think, what could the possible reason be? Well, it might just be that there's, less glucose because they need it to respire, don't they? If there's so many of them, they're going to be competing for the same glucose, or the same oxygen. Or you could say that they're making ethanol, which is toxic to them. Now it says the following equation can be used to make predictions of the growth of the population using these ideal laboratory conditions. So xt is a population after a certain time, x0 is the population at the start, e is 2.72, and then r is the growth rate, and t is the time period at which this applies. So a population of 2,000 was left for 10 hours. The value of the growth rate was this. Assuming no yeast died, Calculate the predicted size after 10 hours. So write down each quantity so you know exactly what you're dealing with. So this is your initial. They've told you that you have x0 is 2000. What else have they told you? For 10 hours, so we know t is in 10 hours, and this is the time period over hours, so we can keep this 10. The value of the growth rate, r, is 0.5. They want you to find how long the population at 10 hours. So xt, x of 10, would just be you slapping these into the formula. So 2000 times e to the 0.5 times 10. If we do that, we get 296826.3, but I'm going to keep it as the same number of significant figures they've given us in the question, or at least decimal places, which we will go with 3. So 298000. There you go. Scientists investigate the process of succession on sand dunes. They measure the percentage cover of different species of plants on sand dunes of different ages, and some of the results are shown in here. So I like to always independent and dependentize everything I can. It just helps me make sense of what's going on. So the independent is what? They're looking at they're changing what? So they're changing the time, obviously, and the, whatever this is, species. So they're changing both of these, and the dependent is the mean percentage cover. Now what we do is we just have a quick look at this data and just have a good idea of what's going on. So as the sand dune gets older, we can see that for beach grass, it goes up, down, and then it just stays down, actually. Whoops. And then for shrub bunch grass, what happens is after about, 25 years-ish, it comes up and then it goes down. And then after that, we got conifer trees, which come up and then down, and then they shoot back up and they stayed like that. And hardwood trees have just been increasing slowly since. Fine. That looks pretty succession-y to me, because succession is where you have that pioneer species, and then you've got new species coming along, they die out, and this and that, blah, blah, blah. And eventually you get lots of different types and lots of different things that are much different to what you originally had. And I could see that here, that this looks like it was the pioneer species, and then since then it's all kind of, come up. It says here, describe how you would determine the mean percentage cover of beach grass on a sand dune. So what a lot of people do is they'll just kind of like describe like random bits. They'll be like, pick places randomly, do this and do that. You need to say what you would do, right? So if we're looking at percentage cover, we're going to put quadrats down and we're going to count up how many are covered, right? So if we're going to do that, we need to describe how we're going to, decide where in this entire whole ass area we're going to do it. Because there's no practical to put like all of these quadrats in the entire area. It's just not very good. So what we do is we randomly chuck them around and then you get a good spread, right? So then here we need to work out the percentage cover, but just also that the mean percentage cover. So what you're going to do is you're going to find the percentage cover at different places with a large number of, and then you're just going to find the mean. So what you do is you need to first of all divide the area into coordinates and use, obviously you write this in full, I'm just being lazy, a random number generator to get coordinates to sample with quadrats. Then what you do is you obviously count them and then you collect the percentage covers. Then what you do is you use a large number of quadrats. Then to find the mean, all you do is you add up all your percentage covers and then divide by the number of quadrats used. That is it. So the scientists concluded that the results were due to succession taking place. Use your figure to explain why they reached this. It's not really like an evaluate thing. They're just saying what in this figure supports this, right? So if we have a look, like I said before, we see that this beach, whatever it is, beach grass goes up and then down, and then all of a sudden these come up. So first of all, I can say immediately that this is probably the pioneer species. Now, we need to look at what's happening now. So they sort of die out and whatever, right? And then you start getting shrub bunch grass and this and that, blah, blah, blah. So it looks like that, but then we need to explain why that's happening. So yes, there are pioneer species, but why were they there to begin with, and why all of a sudden are new things coming along? So what they must have done is that they must have changed the abiotic environmental factor. Like, for example, nutrient availability in the soils that they might have died to decompose to become. So abiotic environmental factors. And this meant they made the environment less hostile for other species. And then obviously remember, link back to this. So it's less hostile for whichever one came next. So the shrub bunch grass. So shrub, blah, 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 right? Bunch grass. So then we still haven't really talked about, the fact that this is succession, because you've got this pioneer species, other species come along, and then eventually you got the sort of, your big thing there that you've reached the end. When you reach the climax, the end of it, it's called a climax community. So this means that whatever that thing, what was it again? Sorry, the conifer trees represent the end of succession, which is that represent the climax community. There you go. Scientists also investigated how the proportion of sunlight reaching the ground didn't change during succession. Some of the results were shown in here. So again, Just have a quick look, almost dependentize it yourself. They're looking at the changing the age. I say changing, quote unquote, they're just measuring it over time. And then as well as that, they're looking at the percentage of full sunlight reaching the ground. So we can see that as we're going from here to here, it's going down, and then it kind of just stays level at about 20. So it says use figure 5 to explain the results in figure 6. Oh, dang, I just got rid of it. 1 sec. So if we look here, from 0 to 500 years, the light reaching the ground was dropping. So let's see what happened between 0 to 500 here. From 0 to 500 here, we eventually led to just a lot of conifer trees being here. So for some reason, lots of conifer trees means lots of light, is not getting to the ground, so it's blocking it. So clearly, it means that the trees are reducing or blocking the sunlight reaching the ground. That's the only explanation based on these two things that I can give. Then it says, using evidence from figure 6, what can you conclude about the net primary productivity of the sand dunes that are older than 1,000 years, and explain your answer? So if we look here, beyond 1,000 years, what do we notice? We can see that the percentage of sunlight hitting the ground is constant. So just a revisitation of what net primary productivity is. So net primary productivity, we're talking, if it's primary productivity, what it means is it's the primary producer doing the thing, right? So it's not going to be anything like, to do with n equals I minus f minus r, because we're not talking about things that are shitting, essentially. Net primary productivity is the same as saying your gross primary productivity minus respiratory losses. What they do is they can, like plants and anything that can photosynthesize, are primary producers. It's in the name. And what they do is the productivity is the energy available to the next trophic level. So they'll do is they convert sunlight into chemical energy. But the thing that they're going to do to lose some of that energy is respiration, right? Plants still need to respire like every cell in the universe needs to exist. So if that's happening, then all of that chemical energy that's made from the sunlight is some of it's lost as respiration. So what's available to next trophic level is the net primary productivity. However, that's net primary production. Productivity is per unit time as well. So here, what we're saying about things that are over 1,000 years old, well, I mean, the light reaching the ground is the exact same. So that means that their photosynthesizing is going to be the same. But the respiration is always going to be the same. So that means that NPP is the same. So NPP is constant because GPP, which represents photosynthesis in these lot, is constant, and respiration is constant. However, I'm just talking about the actual things living in it, because that's constant. Additionally, what you could say instead is that it's less than, because NPP is lower because there's less light, so there's less GPP, but beyond that point it's constant anyway, even though it is lower. Furosemide and CBT are drugs used to remove excess fluid from the body. Scientists investigate the effect of these drugs on the volume of urine produced by the human volunteers. So what they did is they got these methods. They put them in A, B, and C at random. They gave them all the same food for 3 days. After 3 days, they gave A tablet containing furosemide, B, a tablet with CBT, and C, a placebo with neither. So A was furosemide, B was CBT, and then C, placebo. And what are they doing? So then they found the mean volume of urine made in each group in the 4 hours after taking the tablets. So independentize it, that helps a lot. So the independent. So independent is whether they've gotten, furosemide, CBT, or placebo. And the dependent is the mean volume of urine 4 hours after. That's what they measured, right? So be specific to this example because it will come back later and be relevant. Some of the results are obtained in the table 1, and a value of plus or minus 2 standard deviations from the means includes 95% of the data. Fine. Let's have a look what's going on real quick. So in A, the mean volume of urine produced in 4 hours was highest, followed by B, and then followed by C. And also we've got our plus or minus 2 standard deviations. These two don't overlap, and nor do these. So they don't overlap on first glance. So it says all the volunteers were given the same food for 3 days. Suggest and explain one reason why they were all given the same food. So whenever you do these kinds of questions, link back to their independent and dependence. Will this screw up the dependent? So if we're giving them the same amount of food, that could be done to mitigate making either this go higher or lower, the mean volume of urine. So you need to think of a possible link to that as to why they would control that. Remember that anything that isn't you changing, furosemide versus CBT versus placebo needs to be exactly the same as much as possible. So in this case, they've told you that they've given the same food, so they're controlling the food. You need to think, why can that screw up the amount of urine they produce? Well, if you eat food, you're either going to put more or less water in your body, so you're going to make more or less urine. Urine is made, its volume is dependent on the amount of water in your body. So clearly, the reason it must be is because it affects body water potential, and hence it can affect the volume of urine, their dependent variable. So it says, using table 1, what can you conclude about the effectiveness of furosemide and CBT in the removal of excess fluid from the body? So it's saying, what do you think about furosemide and CBT in getting rid of fluid from the body? So here then, we could see that A is the best, furosemide is the best, followed by CBT, followed by the placebo. So A and CBT are better than the placebo, with A being better than CBT. And also, as well as that, like I said before, there's no overlap in their standard deviations, so they are pretty effective based on these results alone. And that's what they want you to do, is only on these results can you say these are effective or not. So then we would say furosemide, blah, 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 and CBT are better than the placebo, and furosemide is better than CVT because they make increased volume of urine, right? Which would help remove excess fluid because you're peeing it out. And then also, as well as that, no overlap in SDs, so their differences are significant. Furosemide is sometimes used to treat high blood pressure. Suggests how furosemide would cause a decrease in blood pressure. So the question's told you, furosemide, what it does is it makes you pee out. If you're peeing more, you're going to have less water in your body, and blood pressure is all to do with how much volume of blood you have. So the volume of the thing it's flowing in, it's how much you can put in there. So a lot of people get the concept of volume mixed up here and pressure. If I have an artery, let's say, for example, here's a fixed size of it, right? If I put more blood volume in this, I'm putting more blood inside it, right? That's going to increase the pressure because this is the overall system's volume is the same. If I change the volume of the artery, so for example, if I have this vein instead, but I keep the water the same, then the pressure drops because it's almost like you have that same blood pooling like this. So if I have a water bottle and I add more water into it for some reason through a tube that's somehow, like here's my water bottle, right? And it's fixed, but I'm increasing the volume of water. I'm adding more water, the pressure's higher. Whereas if I make the bottle just bigger, but I have the same water, then the pressure's a lot lower. It's the same concept here. So here, you've got lower volume in your lower blood volume because of you're peeing out more. And if you've got lower blood volume, that means there's less of it being shoved into your arteries, the pressure's lower. Furosemide inhibits the absorption of sodium and chloride ions from the filtrate produced in the nephrons. Explain how this would increase the volume of urine produced. So quick crash course on how your kidneys work. So you have your afferent arterioles coming to your nephron, and then a bundle of capillaries called your glomerulus, and then your afferent arterioles. What happens is you've now got your Bowman's capsule surrounding it, and then you've got your proximal convoluted tubule here. You've got your descending limb of the loop of Henle, the ascending limb of the loop of Henle, your distal convoluted tubule. Proximal means nearby. So proximal, nearby, convoluted bendy tubule, tube. And distal means far away. So far away, convoluted bendy tubule. And then you've got your collecting ducts here as well, like so, right? Now, the question's told you about sodium and chloride. Now, many people might not know about chloride. That's fine. We're talking about sodium. And also, as well as that, it inhibits the absorption of sodium and chloride from the filtrate. So it's coming out. Normally, what happens is at the ascending limb, the sodium ions are actively pumped out, right? And what the water can't get back in, sodium can't get back in. The reason is because then that lowers the water potential of all of this surrounding part here on purpose. This whole step is just to shove down the water potential, because then what happens is water can actually leave from the descending limb here. So because we've just forcibly made the water potential lower, water can move out via osmosis, and then it's picked up by capillaries that are sort of, chilling around here like that. That's how we reabsorb water from the loop of Henle. And near at the bottom here, sodium ions can also move out here as well by diffusion. And that again is to continually make the water potential as low as possible here so that we're forcing water to follow, right? Now this question has told you that we are now inhibiting the absorption of sodium and chloride. So what's happening is that step isn't happening, and also along with it, it's told you chloride, right? So if that's not happening, then that means no sodium's coming out and it's staying inside the filtrate, the fluid in the tube. Eventually, it's just going to pass through, blah, 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 and then out into your pee. So then if the sodium and chloride is being passed into your pee, then think about it in terms of water potentially stuff, all of this topic. Inside, there's more sodium and chloride, which means the water potential inside is going to be lower, so the less water is actually going to move out here. It's going to stay in here, and you're going to pee out more of it like that. So that's exactly what happens. So the water potential of the filtrate, or the fluid in the tubule, decreases. So that means you get less water reabsorbed, and you need to say by how? Via osmosis. So therefore, you've got more fluid volume. Sorry, urine volume. And as well, you would also say where this is happening, is it's blah, 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 because less absorption of water at the loop of Henle, specifically the descending loop of Henle. As well as that, the collecting duct can also do it, because water moves out as a consequence of this too as well. But it's mainly the descending limb of loop of Henle and the collecting duct as well. The scientists also measured the mean rates of the flow of blood plasma to the kidneys. The scientists' results are shown in table 2. So if with furosemide, you've got the mean rate of blood flow to be 380, and then B, it's a little bit lower, and then placebo, it's lower as well. So what's happening here is with furosemide, there's more plasma going to the kidneys versus in CVT, and there's less than placebo. The mean rate of blood flow plasma is 60% of the mean rate of blood flow into the kidneys. How much greater is the flow of blood into the kidneys with furosemide than with group C over the 4 hours? Give your answers in centimeters cubed. So for A and B, per minute, it's 380 for the plasma and then 295 for the placebo. So we want to do it for 4 hours. We're going to do 380 times 4 times 60 because we want to do how many minutes for 240 minutes, right? So that gives you 91200, and then for this one it's 295 times 4 times 60, which gives you 70800, right? But that's the plasma. The question's told you that the plasma is 60% of the blood. So what you would do is you do 91200 divided by 0.6 to get the original back up. So it would be 152000, and then 70800 over 0.6, which gives you 118000. The difference between these two is 152000 minus that gives you 34000 centimeters cubed. There you go. In genetic crosses, the observed phenotypic ratios observed in the offspring are often not the same as the expected ratios. Explain why. Like, dude, like, you know, if you have like a parent with blonde hair and then somebody else with black hair, you know, not every single person will appear as a nice 3-to-1 ratio of blonde, you know, black hair to blonde hair, right? Like, you know this. So then we need to think about why in genetics terms that's not the case. Well, one reason, first of all, is that, I mean, no one's having like 10,000 children to be able to get that ratio to fit. You'd only have like 2 or 3 or 4 or 5. So small sample immediately it's what's coming to mind. Small sample size. But the other thing as well is that it's assuming that all of these, you know, gametes, once they're breeding and stuff, are going to just fuse exactly in the same pattern to give you a nice 9331 ratio, which is not the case. Fertilization, infusion of gametes is random. As well as that, you can have sex linkage, for example, where it's on the X chromosome and that's passed on. And if you don't have like the corresponding Y chromosome, so like, Y chromosome is shorter than the X, right? So if you have an autosomal recessive thing here, then it means it's going to be expressed because there's no other X to like, you know, combat or whatever. So that's also one. And then as well as that, you could have something like epistasis. So the expression of one gene masks the another one. So for example, if I have like, I don't know, this is my black hair and I have the black hair gene, but I also have another allele, sorry, not gene. I have another allele that makes me bald. The bald allele is masking the expression of the black head allele regardless. I might have the black head allele, but I got no hair. So like, you know, it's being masked by the bald head gene, so it's epistasis there. In tomato plants, the genes for height and the type of leaf are on the same homologous pair of chromosomes. The allele T for a tall plant is dominant to little t. So I'm going to write that there. Tall, little t is dwarf. Big M is normal. Little m is mottled. Now you've got to be like really, really strange if you're not doing this. So then it says a biologist carried out crosses between parent plants that are heterozygous for both genes and examined the offspring produced. The position of the two alleles for both genes within the same in each parent as shown in this figure. The phenotypes and the number of offspring are produced are shown in this table. So here we can see that the gene is found at this locus on this chromosome, and these are the two alleles, and this is the gene for whatever the leaves thing here, and these two alleles at the same locus here. Now, the phenotype of the offspring here are tall plants and normal leaves, tall plants and mottled leaves, and then dwarf plants and normal leaves and dwarf plants, mottled leaves, whatever, right? So we've got mainly more of these and more of these, fine. And as well as that, let's just quickly scribble down what the parents originally were. The parents are heterozygous for both of them, so it'd be big T, big M, little m. Now it says, what would the genotype of the offspring with dwarf plants and mottled leaves be? So recessive, and you can have two chromosome, so little t, little m, little m. So now it says, use the information to explain the results in table 3. So what we need to do is just have a quick look at the ratios of all of these, right, as well. So if I do, we're going to have 1860 to 68 to 57, 57 to 580. You divide by the smallest number first to get a ratio, so you're going to divide them all by 57, right? So let's see what we end up getting. So 1860 divided by 57 gives you what, like 32.6. That's going to be roughly, what, 68 divided by 57 is going to be like 1 point something, right? 1.2, 1, and then 580 divided by 57 gives you what, 10.2. That's not like 9331 that we like. We can see there's more towards this side and less towards here. Let's have a quick look at what phenotypes would make this. So if we see here, if it's tall and normal, our possibilities are that you're going to have big T, big M, or it could be big T, big M, little m, right? Those are the main ones that would give you this. Now the possibilities that make tall and mottled would be big T, big T, little m, or it could be big T, little t, little m, little m. And then for dwarf plants and normal leaves, it'd be little a, t, big M, or it'd be little t, big M, little m. And then for dwarf plants and mottled leaves, it'd be little t, little m, and that's it. So clearly, based on this cross, what must have happened for them to mainly be here and here, the parents must have made more of these and these, right? So the parents must have made big T, big M gametes, or little t, big M gametes, or little t, little m gametes mainly. Otherwise, you know, why on earth would it be this? When that's happening, where you have this unexpected ratio that doesn't fit 9331, you need to be thinking, is it something to do with epistasis, sex linkage, or autosomal linkage? Here, they're making more of these, so it has to be autosomal linkage because these are quite close together. They must have just been passed on. So if we take, for example, a pair of homologous chromosomes here, for example, this, right? And in meiosis, they're going to replicate first and form these. Homologous, remember, is just maternal, paternal, like parent 1, parent 2, maternal, paternal, paternal. It's not maternal and paternal. The homologous pairs just look like this because they're doubled and copied. What might happen is during crossing over, for example, here, you can see that will happen. Let's pretend we have two genes here in these two alleles. Because they're close together, they're not being crossed over, so these will get passed on anyway. So for example, if that's paternal and paternal, then the eventual gametes were more likely to have these two as the same as the original parent. That's what autosomal linkage is. It's where these genes that are on the same autosome are close together, they're linked, so they stay together when you're passing them on to the offspring. So clearly what's happened here is that these alleles must have been made more simply just because there's less crossing over happening in this sense. So clearly, these genes, first of all, are autosomally linked, and the parents produced fewer gametes, so fewer main, and we need to describe them really. So like, they've made fewer, what's that, big T, little m. So tall plus mottled or dwarf and normal leaved offspring, gametes, must have been because these alleles were passed together during crossing over. That's what autosomal linkage is. Sex linkage is where, for example, you have like a Y chromosome, which is short and mashed up compared to the X. And what happens is you can have like a recessive allele here, but no corresponding, dominant allele to mask it. So men might have, for example, sex-linked recessive diseases like colorblindness. So now it says, complete the table to show expected ratio of phenotypes of the same cross have been carried out, but the same genes for height were on different homologous pairs of chromosomes. Because they're not on the same chromosome and next to each other or anything like that, the likelihood of linkage, autosomal linkage, is going to be lower. So what we're going to get is we're going to get that lovely 9331 ratio that we're familiar with, right? But we need to actually look at what they would be. So these 9331 ratios would be, well, it would be tall and normal, tall and mottled, dwarf and normal, and then dwarf and mottled. All the recessives, mixture of the two, and then like all of them being big T, little t, or, or big T, little t, or big T, or big M, little m. And you would get your nice 9331 ratio. Dopamine is a neurotransmitter released in synapses in the brain. The transmission of dopamine is similar to that of acetylcholine. It stimulates the production of nerve impulses in the postsynaptic neuron. Describe how. Do not include the events leading to the release of dopamine and the events following the production of nerve impulses. Right, but just for the sake of teaching you guys how it works, I'm just going to quickly talk about the events beforehand, but we don't need to talk about it in the answer. At cholinergic synapse, what happens is, so synapse is a connection between a gap, really, but it's a connection between a nerve and another nerve, or a nerve and an effector like a muscle or gland, right? This gap is called your synaptic cleft. This is your presynaptic, before the synapse membrane or neuron, the whole thing. This is your postsynaptic membrane or whatever it is, or neuron, so here, right? What happens is you have an action potential arriving in the presynaptic nerve, which then causes calcium ion channels to open. So CO2 plus ion channels open because they're voltage gated. They will only open if the voltage is right. And then what happens is they'll get in, and then they cause vesicles that contain neurotransmitters to fuse with the membrane like this. When that's done and dusted, the calcium 2 plus is actively pumped out, and because we don't really want to keep it in here, it will keep doing that. What this does is it releases your acetylcholine into the synaptic cleft, and then it will bind to complementary receptors on the postsynaptic, the aftersynaptic membrane, right? The whole thing with depolarization is to do with sodium ions moving in. So if that's the case, this leads to sodium ions entering like this. These are ligand-gated sodium ion channels. They only open if this is met. And then you get a generator potential because you get depolarization here. If the threshold's met, then you get an action potential like that. Once the acetylcholine is done and dusted with its business, it's then broken down by acetylcholine esterase, and then its products are reabsorbed in the presynaptic neuron so it can be remade. The reason is because if it just kind of stayed here, it would keep stimulating this, and you'd keep getting action potentials here, and that can cause problems. This is called an excitatory inhibitor because it's causing a depolarization here. Sometimes you can have it where the ligand binds here, for example, GABA, and it can lead to things like chloride ions moving in here. That means the inside's going to be more negative, so it hyperpolarizes this, making it actually inhibitory and harder to cause depolarization. So that's just a quick rundown on how these synapses work. So back here, they just want to know how it actually works once it's been released. So then what happens is it's the same thing. So the dopamine, I'm just going to write dope just because I'm lazy. You write the whole thing. It diffuses. You can't just say moves. You need to explain why. High concentration, lower concentration. Across the synapse. And like I said before, it binds to complementary receptors. But you need to say where. On the postsynaptic membrane. Then what that does is it opens Na plus ion. You can't say sodium. It has to be sodium ion channels. We don't have solid sodium metal in our blood and fluids and stuff. And then it leads to depolarization in the postsynaptic neuron. The reason that you need to make it clear that it's presynaptic and postsynaptic is some neurons, what they might have is they can have receptors for the neurotransmitter on its own thing as well. So what could happen is you can have the postsynaptic neuron with these receptors, but it can also be on the presynaptic one. The reason is it might be like a negative feedback sort of thing where it binds to this receptor and then it calms down the release of it like that. So it's a self-regulating thing. So it's really important you make it clear which way things are moving, from where to where. General rule in biology, if you're saying something's diffusing, you need to say from where to where. And if you're going to say something, you can't just say something, you have to say what it is. You can't just say it diffuses. If you're saying it or they or something, you don't know what you're talking about. Be direct, because your examiner is not going to sit and read all of your its and theys and try and work out what it is. If they can't be asked working it out, they're probably just going to leave it. So please just say what specifically is doing what. Dopamine has a role in numerous processes in the brain, including pain relief. The release of dopamine can be stimulated by chemicals called endorphins produced in the brain. Endorphins attach to opioid receptors on the presynaptic neurons that release dopamine. Morphine is a drug that has a similar structure to endorphins and it can provide pain relief. Explain how. The question has told you that it has a similar structure to endorphins. Endorphins attach to these receptors and then it leads to dopamine release. So you would just say that morphine does that. So morphine clearly binds to opioid receptors, which means you get dopamine release. You can't start yapping about active site and this and that, blah, 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 because active site is only to do with enzymes. There is no enzyme here, no active site. It's a receptor. Oh my God, as if on cue. Like I forgot that this was here. So GABA is a neurotransmitter released in some inhibitory synapses in the brain. GABA causes negatively charged chloride ions to enter the postsynaptic neurons. Explain how this inhibits postsynaptic neurons. So remember how I was saying before in the previous question when I was explaining the synapse, this and that, blah, blah, blah. It binds to the receptor here, and then you get these nice Na plus ions coming in, right? In this case, they've told you in the question that when GABA binds to a receptor, it causes negatively charged chloride ions to enter instead. So now what's happening? The inside of this is becoming more negative. If that's more negative, then if I want to cause an action potential, then I'm going to have to shove way more Na pluses in here because the membrane is hyperpolarized to get to that threshold and then bang, right? So that's what it essentially is. And that's like I said before, it's called an inhibitory neurotransmitter. So what happens is now when this postsynaptic neuron becomes more negative and hyperpolarized. So then what happens is you need more Na plus to cause depolarization and then hence an action potential. The reason it's called depolarization is because the inside of the nerve is negative compared to the outside. If sodium's moving in, that's making it more positive, and they're now becoming more and more similar to each other and equal, or it might overshoot. This is called depolarization because there are poles here. There's a positive and a negative. If you're depolarizing, you're making this pole disappear. Hyperpolarizing means you're making this pole, positive and negative, more wider. So the inside's even more negative, then the difference between the two is higher. So it's just something that I've realized that a lot of people really don't get for some reason. I don't know why it's not made clear when teaching it, but here it is. What is meant by a genome? It's all the DNA in an organism. So chromatin immunoprecipitation is a method to determine where a transcription factor binds to DNA. The principle behind this procedure is shown. So you add chemicals that bind to transcription factor to the DNA. You break it into small fragments and add an antibody specific to the transcription factor. Then what you do is you remove the precipitated DNA and then you add a chemical to remove the antibody with the transcription factor, and then you amplify the DNA which corresponds to the site occupied by the transcription factor. Fine. So then it says here, explain why the antibody binds to the transcription factor. The only reason an antibody would bind to anything is that it's complementary to whatever it is. There's no active site, so please stop saying active site. If I see that, I'm going to kill you. So then what happens is, that's a joke, I'm not going to kill you, by the way. The transcription factor must be complementary to the antibody binding site. That is literally all you need to say, because it's the only explanation to say why it does that. Then it says, use figure 8 to explain what precipitated DNA consists of. So here, where are we? So we add transcription factor to the DNA, we break it down, we add an antibody, and then we get precipitated DNA. So this clearly must be that there is DNA, the transcription factor, and then the antibody. That's all there is. It says ignore reference to chemicals. If you're saying chemicals in this, dang, man, what are you doing? Soybeans. Oh, no, bro, they put in soy in these questions. Oh, no. That's a joke, by the way. Soy's pretty good. Please stop being scared of it. It's not going to kill you, promise. You don't have to be all like alpha male going out and like beating up like bears in the woods. Relax. None of that's needed, dude. Don't got to be doing all that. Soybeans are used in a number of processed foods. However, soybeans contain a protein that's called P34 that causes an allergic response in some people. Scientists have created a transgenic soybean that produces single-stranded cDNA, which prevents the transcription of the P34 gene. They then use recombinant plasmids as vectors to transform soybean cells, and after they'd screen these cells for production of this P34 protein, they culture the transformed cells to form soybean plants. Suggest how single-stranded cDNA can stop the transcription of this P34 gene. So then, think about it step by step. You've got P34 gene. The entire point of transcription is to make mRNA. And the question has told you is how this could prevent the transcription of this. When you're doing transcription for any gene, you've got the DNA, blah, 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 you break open the hydrogen bonds between these to expose bases that you want to make mRNA with, right? So then your free floating RNA nucleotides join together, RNA polymerase joins together, and then you've got your mRNA here. The question's told you how this single-stranded DNA could stop this thing happening. So maybe at this exact point, you've got the single-stranded DNA that comes along and then just plunks here and then forms complementary base pairs instead and stops it from happening. So that's exactly what it is that it binds to the P34 gene to prevent mRNA transcription. It says here, reject transcription factor. The reason is because it literally says to stop the transcription of this gene. And it's single-stranded DNA. So it's not going for the transcription factor, it's going for the DNA itself. It's hinted at in the question. Now it says, describe the roles of two enzymes that can insert DNA fragments into plasmids. So a fragment of DNA literally is just a piece of DNA with whatever you want in it, right? If we want to put it into a plasmid, what we need to do is we need to chop out a space for it, and then we need to stick it in. So we can chop out a space for it like this, but then you can just slap this here and it's just going to sit there and maybe over hours join. We can use something to speed up that process to join them like this. So what we do is we use something called restriction endonucleases to cut them, right? Because what it will do is it will create DNA like so with sticky ends here, like that and that. These are just bits of unbound base from the other strand that's missing. But if you use the same restriction endonuclease here, what it'll do is it'll find wherever it can recognize, and be complementary and blah, blah, blah. And then it will create the same sticky ends here that you can just slot this in with, because the sticky ends from this one will be the same for T here if you use the same restriction endonuclease. So then what you would say is the first enzyme is you'd use the restriction endonuclease. And the entire point of it is just to cut the plasmid. You need to say in this context, you can't just say it cuts DNA. We're talking about how you're going to deal with this, right? So it cuts the plasmid and there's space. Now, the other thing that we can use, like I said before, is we need to actually slap it in here and let it do its thing. An enzyme called ligase is used. And what that does is it joins the DNA, the fragment, to the plasmid, like that. The soybean cells are screened for the presence of the P34 protein. This involves using gel electrophoresis to separate proteins extracted from soybean cells. Suggests two features of which the structure of different proteins that enable them to be separated by electrophoresis. So gel electrophoresis, when you're doing it for DNA, what you do is you put it in a well here, you add a solution that's conductive, and you slap it into the gel, whatever, blah, 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 and then you set up a positive and a negative electrode by putting a circuit around it. And then what happens is, because DNA is negatively charged, it will move, by the way around, whoops, that's supposed to be negative, that's positive. It will move away from the negative towards the positive, right? And if it's heavier and it's bigger, it will move slower, and the lighter ones move further. So it separates them based on their length, right? So here it says, what features of proteins let them separate them by electrophoresis, right? So if electrophoresis in the context of DNA is separating them based on their length, it's probably going to be separating them based on their length here in proteins. So we need to think, what determines a protein's length? Well, it's clearly, for example, one of them is definitely the amino acid sequence length, as well as that you could talk about their R groups. If they have really fatty R groups, they're going to move slower. But then that only tells you why they're able to actually, form separate bits. But what if it doesn't move to begin with? It's saying how the structure of these proteins lets them actually do this. It also must mean that it has to be charged as well. For example, it could be negative charge or positive charge. If you don't say this, then there's, when you're doing electrophoresis, it'll just stay here because it's not charged. It's not bothered by the fact there's an electric field around it. So you have to include that too. Right. So lactose is the main sugar in milk and hydrolyzed with the enzyme lactase. Lactase is essential to stop newborn mammals as milk is their only source of food. Most mammals stop making lactase and they start feeding on other food sources. Humans are an exception to this because some of them continue to make lactase. The ability to continue producing lactase is lactase persistent, and this is a dominant allele. So if you have lactase persistence, you're still making lactase, even though you don't. You're supposed to not, right? So you're still making lactase, and this is a dominant thing. And then a number of hypotheses is based on different selection pressures that have been put forward to explain LP in humans. One hypothesis for LP in humans suggests that the selective pressure was related to some humans farming cattle as a source of milk. Describe how farming cattle as a source of milk led to an increase in LP. So everyone I've seen a lot of people just dive into this when they brought this question saying, bro, I said the survive, reproduce, blah, 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 blah. Model answers do not work. You just can't do that. And it really, really gets on my nerves because you guys just yap about all kinds of garbage and I've seen it. It just does not work. When you're talking about, I'm just going to give a quick side crash course. In a population, we are not all identical because we have different alleles and those alleles came from mutations. That is called variation. Now, some variation can lead to advantages over others, depending on what scenario you're in. This means that if you have this advantage, then yes, you are more likely to survive, right? You're more likely to survive and reproduce and whatever. And then over time, you're going to pass on this allele. And those that have it will also survive and reproduce, blah, blah, blah. And you're increasing the frequency, the number of times that allele appears, right? Now, natural selection is this entire process here where what happens is the individuals that have this advantageous allele are able to survive, reproduce, blah, 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 blah. And the types of selection, the reason this happens is because there is a selection pressure of some sort. The selection pressure is basically saying you are allowed to survive and reproduce, you are not, right? And over a lot of generations, you eventually get evolution as well. Because as you get more and more frequency of this allele, you get a gradual change of this species over time. And that's what evolution is. So selection pressures are what's driving this process. So, for example, if we have rabbits used to be small-eared as well, and then one geezer had a mutation and had some variation that gave them big ears. That big-eared one had an advantage over the other smaller ones, so it was able to survive, reproduce, pass on the allele, and over time, the ones that had the small ears weren't able to survive and reproduce as much, and then eventually get an increase in the frequency of the big ear allele. So the reason is not that this selection pressure was because of big ear or little ear, is because the selection pressure in that context was that they could detect predators more easily. Selection pressure is the thing specifically that's causing this differential, as in the reason it's causing the differential survival, as in causing the reasons, or sorry, I should say causing / the reasons why some survive over others, right? This selection pressure means that you're more like, the ones that can cope with it and deal with it, survive, reproduce, pass on, blah, blah, blah. The types of natural, so that leads, those are because of different adaptations, blah, blah, blah. The types of natural selection you get are directional. So directional is where individuals towards an extreme, so for example, like, I don't know, height, right? This is the number, are favored. So generally speaking, this is your population. Most of the people are in the mean, right? So then directional selections means that either your littlest ones are the most likely to survive and reproduce, or, the tallest or whatever it is. Either extreme is favored, right? So there might be some sort of selection pressure that means that those towards each extreme are fine. The next type is stabilizing. It's in the name. Stabilizing means that it's stable, it's nice and happy. Those towards the middle are more likely to survive and reproduce. For example, human birth weights, if you're very, very underweight, you're less likely to survive. If you're more and more too heavy, you're less likely to survive. Those in the middle survive. The other type that you have is also disruptive. Disruptive is where either extreme is favored, but those in the middle are not, right? So when you're doing these styles of questions where it's talking about an increase in frequency of alleles over time, clearly there has to be some sort of selection thing going on. It could be artificial selection where, for example, if I have a load of plants and I have tall plants and little plants, and I only let the tall plants reproduce, then the tall plants are surviving and reproducing, blah, 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 passing on the alleles. But the selection pressure is because I'm saying tall plants are allowed to survive and reproduce, small ones are not. So don't jump into a model answer like this, step by step it. Here's the thing, here's what this leads to, then this, then this, then this. Because there's some questions where it has shown, like, scientists, for example, only bred this thing versus that. And then people are saying, oh, they're more likely to survive, reproduce. Like, what? By what? They're living in a lab. They're being fed everything they want. They're happy. So you just be careful with that, right? So this question has told you that for some reason, you have farming cattle as a source of milk, and then you've got increase in LP, which is an allele. So if you have an increase in this allele, what you must have is you've got evolution, which probably came from some sort of selection, because why on earth would it lead to an increase? If it didn't matter, we would all have it. We'd all be the same. We'd all have no LP. But clearly, this thing in this scenario here, this environment for these people is the same, because otherwise, every human in the world would have no lactose, intolerance. We'd all be dead, and that's not the case. So you need to be careful to relate it to the environment they are in and what's going on. So how has this led to an increase in LP? Well, first of all, the variation, the fact that some have LP and don't, has to be due to the mutation. So the LP allele comes from a mutation. So it arises from a mutation. And obviously they've told you in the question that this is a source of milk and this is lactase, which, hydrolyzes lactose. So clearly they've got mutations. So some individuals are able to digest lactose from milk versus others. The ones that can obviously now have more nutrients. You need to explain why they're able to survive and reproduce. You can't just go, survive and reproduce, blah, blah, blah. You need to say why. So therefore, the ones with the LP are more likely to survive and reproduce. And over time, because they're the only ones more likely to survive and reproduce, over time, the LP allele frequency, how often it appears, is increasing, right? So you're favoring an extreme where you can make LP versus not. So what you've got here is a specific type. You've got directional selection as well. But that's the bit that I see people fumbling, where they jump straight to a modern sort of model answer. It don't work because, it just doesn't make sense. You've got to relate it to the context here. And it's not a gene either. A lot of people say passing on the gene as well. Sorry, advantageous gene. They both have. Here's the chromosome for one person, here's another. The gene is simply just where it is found. LP, no LP, this is the LP gene. The gene is still passed on, whether they have LP or not. It is the allele that's different, and the alleles are the base sequences that relate to them. If you have an LP here, you might have some base sequence, and if you have no LP, the base sequence might be different somewhere else. It doesn't matter because they still have an LP gene. One just doesn't make LP, one does. So it's important that you say allele, which are different versions of the same gene. Use the information to explain why people who have LP would rapidly increase once selection had been established. So it's telling you that at this point in time, selection for this thing, so as in these people with LP are more likely to survive and reproduce. Why is it happening fast, right? The only reason I can think of, based on all of this information alone, is it's got to be something to do with the allele being passed on. And what, right? If we look at the allele, it is dominant. So that's the only reason as to why it would go faster, because it's clearly that it's just dominant. So LP is dominant, which means that it's always expressed, even with one copy. And that makes sense, because if you had to have two of them, then very slowly, yes, over time, if you had both of them, you'd survive and reproduce. But here, you just need one and you're sorted. So lactase persistence is called mutation in the gene. This does not occur in the gene coding for lactase. So the rules are you may have a gene, you transcribe it into mRNA, and then you translate it into a protein. That's the entire system. Now this question, remember that lactase persistence is where you're still making lactase. Remember, that's what it is. So clearly, we're still transcribing and we're still translating, but there's no mutation in the gene. So something is happening somewhere else that is a mutation that's leading to transcription translation still happening. So clearly what might be going on is that, for example, we might have, remember proteins, so when you want to encode a gene and express it, you have regions before it of sequences called promoter regions, right? And they say, hey, it's time to get going, let's go. And it causes the transcription of that when the transcription factors bind to this. It's told you in the question that over time, it's not expressed anymore. So maybe that these promoter regions are still chilling, or there's a mutation in them to stop them to keep them still working, if that makes sense. It's not that it's not working anymore, it's that it's still working. So what could happen is we have a mutation in this the promoter, and the transcription factor still able to bind to it, which means you get mRNA and protein, or it could have a mutation in the transcription factor itself. So what happens is it might change over time and then not work, but then for some reason it's still working. So I'm going to choose either one of these. I'm going to just go ahead and say mutation in the promoter. You still get lactase production. So you still get lactase is transcribed and translated. The whole topic is about the bridging of genes and the proteins, which is going to involve transcription and translation. All of these topics, when it comes to expression, is talking about things screwing up or changing transcription and translation. I've seen a lot of garbage advice from people on YouTube. Some individuals I'm not a big fan of, to be honest. But there's no way of going around this. Skim through the passage, make sense of what's going on, look at the question and what the question has told you as a fact, and then go back. What people do is they read a question and they're like, I think this is testing me on this topic. I'm not going to blurt out all of this garbage for nothing. Or they just repeat what the text says back to them. You read it, you establish the facts of the question, and then you come up with an explanation why. It's almost like treating every single thing that says explain as suggest instead. I've heard a lot of garbage from individuals who are on YouTube saying all this garbage advice, and I end up having to see them when I'm teaching it, and it drives me absolutely bonkers because, like, it just don't work. So complete achromatopsia is a form of complete colorblindness, and it's caused by having only rods and no functional cone cells. People with complete achromatopsia have difficulty seeing and difficulty. Complete achromatopsia is caused by an autism or recessive allele and is usually very rare in populations, with only 1 in 40,000 being affected. However, on the Pacific Island and Pinch Lapid, 10% of them are affected. One form of red-green colorblindness is caused by sex-linked recessive allele, which affects more men than women. People with this red-green colorblindness are unable to distinguish between red and green, and also between other colors. They have green-sensitive cones, but the photoreceptive pigment they contain does not function. Scientists investigated the use of gene therapy to correct red-green colorblindness in monkeys. They injected viruses containing the gene in the red-green pigment directly into the eyes of the monkeys. Although the monkeys maintain their 2 years of color vision, there is debate on whether the form of gene therapy is worthwhile. So no clinical trials of this procedure have been carried on humans. Current research into the treatment of red-green colorblindness involves the use of induced pluripotent stem cells. The use of iPS cells could have advantages over the use of gene therapy. Use the information in the passage and your own knowledge to answer these following questions. So it's all about establishing what the question and the facts have told you and then explaining it. It says here, people with complete achromatopsia have difficulty seeing in detail. So explain why. So achromatopsia, blah, 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 difficulty seeing in detail. So look at what the lines and the text around it has told you. If you look at lines 2 to 3, which is 1, 2, and 3, people just read that and they're like, I don't know what's going on. People with complete achromatopsia have difficulty in detail. It's just telling you where to go and look. It doesn't mean that this thing here means that's where the information is going to be. You need to go look around it. So if I just went here and I'm like, okay, I'm going to look at lines 2 to 3. People with complete achromatopsia have difficulty seeing in detail. Complete achromatopsia is blah, blah, blah. That doesn't help. Read around it. Read the sentences before and after. So people with complete achromatopsia have difficulty in seeing in detail. Complete achromatopsia is a form of complete colorblindness. It's caused by having only rods and no functional cone cells. That might link to this. So now we've got achromatopsia means, what was it? Sorry. We have only rods, no cones. That's what the questions have told us. Establish the facts first before you dive into the explanation. So for some reason, having only rods and no cones means difficulty seeing in details. Now you dig into your head and say, well, what's going on? So first of all, start by pointing that out. There's no cones, cards, only rods, right? Then we need to explain why that causes difficulty in detail. So then you need to think in your head, okay, rods versus cones. Oh yeah, rods have less visual acuity than cones, right? They have a lower resolution, and we need to think why. So rods have less visual acuity than cones. We need to explain why. That still doesn't tell us what's going on. It's because rods, like multiple of them, are connected to a single bipolar neuron, whereas cones, it's only one. Therefore, that still doesn't tell me why it causes the difficulty in detail. We've said, yes, it reduces the acuity, blah, blah, blah. We haven't told them why. It means that they're not the impulses aren't separate to the brain. Impulses are one, sent as one to the brain. So or you could say, for example, there's separate sets of impulses. What I mean by that is, here's your bipolar neuron. I'm drawing this very, very rubbishly. Just chilling, right? Rod, and here's a thing that you're seeing, right? So there's a nice little spot here. Rod sends impulses this way, and this way to this one singular thing. It goes to your brain, right? And you see just this, because it can't tell apart the two. Visual acuity is poor, its resolution is poor. Whereas cones, cone, here and here, they all have their own bipolar neuron, which then sends impulses separately to the brain, so your brain sees these two, so you see two. So you need to explain that. So can you see how I use this text, use what they told me to then dig into the gaps? Because otherwise it's just rubbish. And you can't say signals either, or messages, or information. You have to say impulses or action potentials. 10% of the population in the Pacific are affected by complete achromatopsia. Use the Hardy-Weinberg equation to calculate the percentage of this population who are heterozygous for this. So what it's telling you, if we go to lines 3 to 6, so 1, 2, 3, 3 to 6, it says complete achromatopsia is blah, 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 blah. It's autism or recessive. So I'm just going to write that there. And it's told you that it's very rare and only 1 in 40,000 are affected. And then it's told you in the Pacific, 10% of the population are affected. So 10% of pop affected. Now, which is 1 in 10, have complete achromatopsia, which I'm going to just call little a, little a. So define what they are, right? So big A, normal vision. Little a is complete achromatopsia. We know that 1 in 10 have this genotype AA. So I'm going to call P big A and Q, little a. So if I know, like, having this genotype is 1 in 10, then the square root of 1 in 10 is going to tell me what A is, because that's the same as saying Q times Q, which is Q squared. So if I know that, then I can do the square root of 1 in 10, which if I do that, we get square root of 1 in 10, you get 0.316. So then we know that P plus Q must equal 1. You can only have 100% of all the alleles possible in a population. So that means that P would equal 1 minus Q, which is going to be 1 minus the answer we just got, which is 0.68377, blah, 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 blah. Now, the other Hardy-Weinberg equation is the P squared plus 2PQ plus Q squared equals 1. P squared is AA plus 2PQ, which could be big A, little a, or little a, big A. That's where the 2PQ comes from. And then Q squared is B is little a, little a. That's 1. They're asking you to find the percentage of this population who are heterozygous. So all you do is you're looking for the 2PQ. So 2PQ is going to be 2 times the 0.316 times the 0.6838, because I'm rounding it. If we do that, we get 0.43216, right? Now that's the proportion, that's not a percentage, that's just a decimal. So to change it to percentage, and I'm going to use the same number of significant figures they gave us in the question, which I believe is 2, it's going to be 43%. That's how you do these. So red-green color blindness affects more men than women, lines 7 to 8, explain why. So if we look at lines 7 to 8, so it says here, one form of red-green color blindness is caused by a sex-linked recessive allele, which affects more men than women, and that's it. So here, they've told you that this color blindness is sex-linked, right? So now you need to explain why it's more common in men versus women. The only reason it could be is that this gene for it, or the allele, must be on the X chromosome. So they need to explain why men have it and women don't. Women, because it's told you it's recessive as well, women need 2 copies to express it. Men need 1 copy of the allele. Because, again, remember, the X chromosome's this, your Y chromosome's that. If they've got this here, there's no Y corresponding X to counteract it, for example. So that's that. People with red-green color blindness are unable to distinguish between red and green and also other colors. Explain why. So if we go back to 8 and 10, so let me just rub this out. It says here that they are unable to distinguish between red and green, but also other colors. Don't just look there and be like, oh my God, I don't know, because it's telling you just where to go to find the text that's relevant, not that you read only those lines. So if we go further, it says they have green sensitive cones, but the pigment they make does not function. So go and establish the facts. Again, they can't distinguish between red and green. They have green sensitive cones, but the pigment they make doesn't work. So then you need to explain what's going on here. So explain why, first of all, they can't distinguish between red and green. Well, it's essentially that. They're not, the green pigment within these green cones is non-functional because the cones are fine. The red, green, and blue cones are fine. It's the pigment within them is not right. Again, work with the question, right? And then therefore, you can't tell red, green apart. There's, no problem. There's problems with red, green. But then we haven't explained why it also affects other colors. Well, if you remember, you only have red, green, and blue cones, but you can see purple, you can see orange, you can see this, you can see that. The reason that is because you're having mixtures of that red, green, and blue. In light, I know this doesn't happen when you're mixing paint, but in light, if you have red, green, and blue combined, you get white light. If you have none of it, you got black. Red and green, in the light sort of realm, makes like your yellows and your oranges. Blue and red makes your purples. So if they're having trouble with other colors, it's because colors are seen based on a combination of all 3 of them. So that's exactly what you're going to say as well. We have 3 different cones types, which are your red, green, and blues. And colors that are seen are due to different amounts of stimulation of each pigment. So different amounts of different cones being stimulated. And multiple of them. There you go. It says current research into treatment of this uses iPS stem cells. If you look at line 17 to 19. So what's that then? 15, 16, 17. So 17 to 19, it says current research involves using these stem cells, these induced pluripotent stem cells. These could have advantages on gene therapy. There's nothing else that's really helpful here. So if we go back here, it says suggest how iPS cells can correct red-green color blindness. So if we're looking at how iPS cells correct this, what do iPS cells do? They're stem cells and they're induced in the pluripotent. So clearly the first thing that they're going to do is they're just going to divide and then differentiate or specialize into green sensitive cones. yes, the problem is with the pigment, but if you make the right cone cells that are genetically modified and blah, 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 to make the right pigment, then that could correct it. Now it says the use of iPS stem cells could have advantages over gene therapy to correct them. Using the information in the explanation in the passage suggests reasons why. So if we look here, what's going on? So the questions told us, again, always work with what the questions told you. They've done it in monkeys, and they maintained it for 2 years. There's no clinical trials of this in the humans. We're looking at iPS stem cells, which may be beneficial, but nothing mentioned about that too. So here then, it's saying, suggest and explain why iPS cells could be better. So we need to say why iPS is better than vectors. So first of all, I mean, the vectors, you only saw 2 years of the color, not having a problem with it in monkeys. It was only 2 years. Maybe iPS would be longer term. You need to make comparative statements of this versus this. So, I saw this and this, but this one, this. You have to make a comparative statement. You can't just list things, right? So then as well as that, if we're using vectors, that involves putting a virus. I think that's what the question told us, if I remember. Yeah. Now, if I inject a virus into somebody, their immune system is probably going to destroy it. If I'm using induced pluripotent stem cells from stem cells of the same organism, the same thing, you're going to get less of a chance of an immune response in iPS. As well as that, clearly if it's 2 years, then you're going to have to keep doing that over and over and over and over and over and over and over again. Whereas in iPS, it's like a single treatment. Once the stem cells are there, it's there. And then as well as that, we're using viruses, which can cause side effects. There you go. So with these questions, right, the strategy is not like, I don't know, just garbaging out or blurting out everything possible. It's just explaining and just really, really narrowing down what is. All of the questions in biology and pretty much every single subject at A level for sciences is, here's a scenario, this happens. Explain why, suggest why. They're not asking you to go and blurt out everything you know about the topic and write a model answer. That literally never works. They don't care. They want to see you take this knowledge and apply it to a new scenario. So all the time, what you have to do is you got to look at the question, establish the facts they've told you. This is better than this happens, this. And then you need to explain using your knowledge why that's happening in this scenario. You could sit and say all about, the treatments of iPS stem cells and this and that in a generic sense and say, oh, well, this, blah, blah, blah. It could become any type of tissue, blah, blah, blah. We don't care about it being any type of tissue, do we? It's talking about the fact that this is going to be treating it here for color blindness and why it's better than viruses. So you're going to answer the question directly. Model answers, in my opinion, just literally never work. Maybe the only time they ever work is those long answer questions in paper 1. But then again, I still think they're rubbish because every exam report you see, it's full of examiners saying, students keep yapping about stuff we didn't even ask. And also, they didn't even address the question and missed out getting loads of marks. Or they said something that contradicted it. So model answers just don't work, dude. If you can explain it in a simple way and then use the right words, then you're chilling. It's not the whole paragraph is the model answery bit, it's the key terms that are relevant here. So for example, if you're going to talk about enzyme active site, you're not going to just say the enzyme is complementary to it. You're going to say the enzyme's active site. And that doesn't mean it's a model answer. It's you showing that you understand how they work, right? Or enzyme substrate complex doesn't have to be a model answery bit. It's you showing that the enzyme and the substrate bind. You can even sometimes paraphrase things as well and you still get marks. So just stop obsessing over it. Don't work, please. Anyway, I hope that helps. Please like, share, and subscribe. You know the drill. And yeah, we'll see you next time.